And now let's turn to part of the major focus for this symposium. What effect does physical debridement have on levels of planktonic and biofilm bacteria using two laboratory models? And the bottom line is that they're very effective at significantly reducing levels of both planktonic and biofilm bacteria. And you're familiar with this product. This is a monofilament-based synthetic pad that contains millions of small fibers, and this helps to engage the biofilm and physically remove it from the surface. So from data done by Dr. Vegan's group, they were able to grow large Staph aureus biofilms, this time on a glass surface, and then using a very precise tangential shearing test using the debrider pad or cotton gauze, you can see that as this moves across the biofilm colonized and grown glass plate, that the amount of biofilm that remains is significantly reduced. And this is a very precise biomechanical model of what happens. And combining this data from Dr. Vegan's group, you can see that the key here is that when you look at the percent of area that is cleansed, in other words, high numbers are good here, you can see that the monofilament debrider, the Debrisoft, is able to remove high levels, over about 90% of the total biofilm surface area, even using 8 sequential new glass slides. So this has a very high capacity to remove the biofilm. That's in contrast to cotton gauze or other cleansing agents that do not have that capacity to remove. So I'm going to show you just one real quick video. This is a mature Pseudomonas aeruginosa biofilm grown on pigskin explants. This is the best model we have in the laboratory for a debridement of a mature biofilm. You can see actually Dot Weir, who's down front here. This is Dot removing the biofilm in the laboratory, and we're highly indebted for Dot to come up and show biochemists how to do that. And basically, when we measure the amount of total bacteria in this case, and this is planktonic and biofilm, in two totally different experiments, you can see that this is reducing about 2 logs of the total bacterial bioburden that is present in these two experiments. Now, if we refine that just a little bit more and we ask not only what is the total bacteria, but especially the biofilm bacteria, you can see that in these two replicate experiments, this is the total bacterial bioburden. This is the biofilm. So there's about 10 to the 5th or so colony-forming units of biofilm. After debridement, the total bacteria, again, is reduced about 1.5 logs, and the biofilm component, which is critical here, is significantly reduced in both of these, about 2 logs. So that's 99% of the bacteria on the surface. What does it look like? Well, this is a scanning electron micrograph showing a mature Pseudomonas at different magnifications. So this is a high mag, and you can see each of the individual Pseudomonas embedded within this large exopolymeric matrix. This is a really dense biofilm. Lower power and lower power and lower power, just showing you how extensive this is on the surface. After the debridement with 2 pads of Debrisoft, you can see that there's almost no bacteria left. Here's a bug. Here are 2 bugs right here. And the key here is that the exopolymeric matrix has been removed along with the bacteria. So in summary, what I hope I've done is explain to you some of the important aspects about bacteria when they're in the presence of a biofilm community, that this self-produced exopolymeric matrix provides extremely high tolerance, not resistance. That's a different term to microbiologists. Tolerance is the ability to just not be killed by something the planktonics are sensitive to. In addition, biofilms are present in a very high percentage of chronic wounds, probably approaching 90%, and that they impair healing by stimulating chronic inflammation, which leads to those elevated levels of protease and reactive oxygen species that degrades proteins that are essential for healing. Debridement, physical debridement with a Debrisoft pad reduces these mature Pseudomonas aeruginosa biofilms, a total of about 3 logs, planktonic plus biofilm. And that really the good news for you is that biofilm-based wound care is only a refocusing of key components that you already know through wound bed preparation, which is debridement and controlling infection inflammation. but emphasizes the importance of the initial debridement to reduce the level of biofilm. So I hope that's set up both what Andy and Trudy are going to talk about, and so I'll ask Andy to come back.